boss. Good afternoon, everyone. Thank you very much for having us. Thanks to Craig and Maureen and all the leadership at OIS for inviting Xequel Bio to present today within the showcase. So hopefully today you'll learn a little bit about this new organization and the impact that we're aiming to bring within the ophthalmic space. You know, bottom line, our mission is very simple. We want to revolutionize the way that the body responds to injury. That's our mission. And that mission is going to be achieved by our proprietary technology platform, which is going to give us many shots on goal. Based on this new chemical entity, alpha-connexin carboxyl terminal 1, otherwise known as alpha-CT1. We have mined this technology in numerous areas, and this mechanism of action has been applicable across a few therapeutic areas, and we have achieved over 110 preclinical studies utilizing this technology, and also 9 clinical trials that have been conducted over 700 patients. So this has been well studied, and we have learned a lot. This company originated out of the Medical University of South Carolina, and essentially they've acted as an academic incubator for many years, and then recently got private equity backed to start transforming the company. We have a comprehensive intellectual property of over 60 global patents. We have a proven leadership team that has deep expertise within commercial and R&D, which is most important. I mentioned we're private equity backed, and the scientists have also earned and very proud of achieving over $27 million in research funding from the Department of Defense and the National Institute of Health. When you look at our proprietary platform, we believe we have robust commercial opportunities within 3 distinct areas where we're currently mining ACT1, dermatology, pulmonology, but center focus for the company as we move forward is within ophthalmology with developing Inexin, which is an ophthalmic solution for corneal wound healing. And we believe in these therapeutic areas, we're targeting high unmet needs that are really a robust, significant market opportunity. As I mentioned, we have a proven leadership team, and I think earlier I stressed on stage as advice, when you have an organization and you're building your team, you want to make sure you make the right selection and you invest in talent. And I'm very blessed to run with this group here with Wes Brazell as CFO. We recently brought on Dr. Michael Brubaker, one of the best CSOs in the business, Linda Davis within administration, the founding scientist, Dr. Christina Grech, who came from the Medical University of South Carolina, and also my colleague who's here today, Tony Tipton, who will be on one of the panels later on. And as you can tell, the breadth and depth of the team has worked across many ophthalmic companies, so I'm very proud of this team. Let's dig a little bit deep into the ACT1, alpha-CT1 peptide, which is a new chemical entity. Connexin 43, a transmembrane protein, is located throughout the body, hence why we're looking at those therapeutic areas. You find it in the eyes, the skin, the lungs. ACT1 is a peptide based on Connexin 43 to promote gap junction formation, reduce harmful inflammation, stabilize tight junctions, and accelerate reepithelialization. Bottom line, ACT1 has demonstrated in all of its work accelerated healing, reducing of harmful inflammation, complete restoration, and the safety and comfort of the product is well established. I'm going to do my best job here to dig a little bit deep into the mechanism of action. You know, it's important to note that connexins are a key component of cellular junctions. There's going to be a cartoon here of 3 boxes. On the left is going to be a healthy state, the middle will be injured, and on the right is going to be treated with ACT1 to give you a better understanding of the MOA. So on the left, you'll see in a healthy, uninjured state, cells communicate with the surrounding space through connexin hemichannels, and the cells actively communicate with each other through connexin gap junctions. And this is necessary to ensure cellular and tissue homeostasis. In the middle, when you have an injured state, hemichannels become activated during injury, leading to the release of harmful pro-inflammatory signaling molecules into the extracellular space. Gap junctions are disrupted, which impede the cell-to-cell communication during this injured state. However, when you provide alpha-CT1 treatment, you see that our peptide is a connexin 43 mimetic peptide that has a unique MOA that causes connexin hemichannels to be sequestered into the gap junctions, resulting in gap junction formation and stabilization. And this will increase the cell-to-cell communication while reducing harmful inflammation. Bottom line is we're taking this technology in NCE and we are working in 3 distinct areas and we're being very selective, okay? Focus today is obviously ophthalmology as we hear at OIS, and our lead program is corneal injury. And I want to show you some of the preliminary work that we did in a phase 1B that was announced with success about 2 months ago. Inexin, which is our treatment for corneal injury, we believe has the opportunity to promote enhanced corneal healing in multiple diseases. And when you look across the board, no FDA-approved therapeutics effectively accelerate corneal reepithelialization and regeneration, and we want to fulfill that gap. When you look at Inexin, our phase 1B results, in 1B we did enroll moderate to severe only dry eye patients with significant corneal injury. We conducted the study using ORA's Controlled Adverse Environment, CAE. We evaluated 3 concentrations, 0.08, 0.4, 2.0, BID, 15 days, 36 subjects. When you look at the primary and secondary endpoints, obviously phase 1B, we wanted to establish safety and tolerability, and I can tell you it was safe. No treatment-related SAEs, no AEs, and all the work we've done, this is a very clean and safe molecule. We also looked at secondary endpoints both on signs and symptoms, and as you can see, we did achieve corneal staining associated with significant improvement at day 15 and improved ODS at day 15, and at some time points, they were statistically significant, which is impressive for our phase 1B. In summary, Inexin targets the underlying mechanism of injury response to promote regenerative healing. It's a new chemical entity. It's etiology-agnostic MOA that promotes gap junction formation, reduces harmful inflammation, and restores barrier function. We're very proud about this technology. Watch us in this space, and look forward to sharing more in the future. Thank you very much.